I don't think there's a single academic who doesn't know what I'm talking about when I'm talking about, for example, frustrations with scientific publishing. A single cell parasite, which is called Toxoplasma gondii, which is famous for being a cat parasite, can reproduce sexually only in the gut of felines. It doesn't do anything bad to the cat, but if it infects a mouse, it changes the mouse's brain somehow so it loses its fear of cats. And then the mouse runs towards the cat and the cat eats it. If you stay happy and you study what you like and just focus on enjoying it, and it's very hard because there are many forces working against it, and perhaps it's okay because, as I said, it is a loophole in the world that people just tell you, study what you want and have fun. So I can understand why the universe is resisting this. But I think that should be definitely the goal, and it's the number one recipe for success.
In this episode of the 632 Nanometer podcast, Mike sits down with Professor Oded Rechavi, Tel Aviv University biologist, C. elegans whisperer, and occasional rule breaker. Oded walks us through how a worm with 959 cells rewired our thinking about heredity by passing starvation memories to its great-grandkids via small RNAs, how hijacking Toxoplasma might one day smuggle drugs past the blood-brain barrier, and how DNA sequencing enabled scientists to understand the provenance of sheepskin Dead Sea Scrolls preserved for thousands of years in the caves of the Judean desert. The conversation goes beyond the bench, covering the possibility of AI becoming reviewer number 3, and Oded's frustrations with scientific publishing, often expressed on his humorous and popular Twitter account.
So C. elegans has 959 cells in the body every time, and 302 neurons. How can they live with a third of their body as neurons? Like if a third of a human body was the brain, it's hard to imagine how that would function. Like how does that work?
Maybe it would function better. I don't know. I mean, it's true that one consideration, for example, might be that the brain consumes more energy. So that might be challenging with, that could be a limitation on our brain size, perhaps. Not an expert on these type of questions, but I think that the real question is how can the worm be so sophisticated with having just 302 neurons? That's the amazing thing. It can do so many things with so few cells.
Okay, so I'm going to go through a number of tweets of yours that I've screenshotted and ask you to provide the context, okay?
I'll try.
I assume I'll put these up in the video, or we'll put them up in the video, but I assume you remember them, but I think you probably remember these by heart. So, but first question before I do that, how did you get into memeing on Twitter? Like, how did this happen to you initially?
Totally by accident. I think I joined Twitter. Now it's also Blue Sky. Around 2016, I started tweeting. I had a new paper. I wanted to promote it, and it didn't go so well. I thought I don't understand the platform. And then I started making stupid jokes, and people liked them. So I just continued. It's not really voluntary. I just have to do it.
Okay. All right. So your pinned tweet is how your email finds me. So you have a whole series of how your email finds me tweets. This is how I originally, got exposed to you, is through Twitter, and I've always enjoyed these. I think probably for Israelis, it's a very weird American thing to say, like, I hope this email finds you well. It's a strange cultural mismatch, and even for me, it feels strange. Even though I grew up in America, and it's how your email finds me, and you have this is most likely at Technion, where I spent almost 2 years at Technion. There's a wild boar laying on a mattress in a pile of trash next to a garbage can. It's very hard. I mean, I'll put it up, but explain this to me. Like, what does this mean?
So first of all, you're right. This is from Haifa. So this is really where the Technion is, where they have lots of wild boars. And, in fact, I think that I captured the same picture a couple of times, but the one that really caught was the one with how your email finds me, which is, you always get asked how the email finds you, and emails always finds you. You always prefer not to be found by an email, I think. So this sort of series of tweets is about. I have another one where there's some creature hiding in a shell or something.
I have it. That's my next one. Yeah. It's it's like a snail or something, but its eyes it's got the eyes that can articulate, and it's looking out of the shell, kind of uncomfortable.
Yeah, so this is one of the, you're right, it's kind of a very not Israeli saying. It's a little bit like, how does this one go? Good for you. You know that people say in America, good for you. In Israel, like if you tell someone, if someone tells you something good that happened to them and you'll tell them good for you, it's an insult. It's like it doesn't sound like you're trying to be nice to them. So how your email finds me is kind of the same.
There's a Boston or like New England version of good for you that's even stronger. It's something typically women say to each other, but they say, I love that for you.
Wow, that's
So
very aggressive. Yeah.
I love that for you. It's something like my wife, when we moved to Boston, like she was like super surprised by this saying. Okay, you have another one that's another series called correlation versus causation. Mm-hmm. Why do you think? I mean, I think this one's kind of self-explanatory. Why do you think this one gets so much interest? Like it's 6,000 retweets, on one of these. What's the reason that these are so popular?
So you say they're self-explanatory, but I think this is the one cognitive tool that many people are missing. So that was obvious in COVID times, but not only in COVID times. I think that in general, what most people, and at least in Israel, they didn't teach that when I was in high school. They didn't teach the two basic cognitive tools for scientists. One is that not to confuse causation with correlation, and the other one is that you need controls. So this is like obvious things, but I think people are very vulnerable to confusing the two.
Yeah. You have one that actually includes a control. It's like the two seagulls.
I don't remember. But okay.
Okay, and then you have a whole series on kind of frustration. I would call it like these I would call like you have some of them are like science tidbits, and the other ones are like frustrations with academia. Mm-hmm. So you have like one of them is the classic meme of like Warren Buffett and some other a bunch of like older white guys laughing, and they say you quote they pay to publish their own papers and to read them.
Right.
And then you have a few others that are kind of in the same vein. What are you trying to express there? And again, why is this so popular?
This is actually very close to my heart because I think, we'll talk about the science later, but we do lots of things in the lab, and that's all nice. In the science itself, it's a pleasure to do. But the process of science is really slow and frustrating, and scientists connect with that. I don't think there's a single academic who doesn't know what I'm talking about when I'm talking about, for example, frustrations with publishing, with scientific publishing. And this is, so publishing might be the most extreme thing that is really crooked and doesn't work as it should. And this is also something that I'm really trying to improve. And I'll be happy to talk more about this, but this is one of the things that excites me the most is trying to improve the way we publish science and do science.
If you had to break it down, what are the most frustrating components?
I think that probably publishing is the most frustrating.
Of publishing.
Yeah. So that's a hard competition, but I think that the reviews, the fact that the incentives. So first of all, it's extremely hard for the journals. I don't necessarily blame the journals. I think they're far from perfect, but I think that just the way that scientific publishing is structured, that scientists are supposed to review each other's work, and no one has the time, and the incentives of people are often not aligned.
Mm-hmm. You didn't cite this person, but that person turns out to be the reviewer.
Yeah, so it's very common that the reviewers ask you to cite their own work. And of course, they are anonymous, and it depends. The quality of the review and the type of feedback you get depends on what they ate for breakfast and whether they like you or don't like you, or whether you step on their toes or not. So it's really a problematic system. People always
say... But isn't this like complaining about gravity in a way? Because like if you get rid of the reviews, there's archive, right? So in a way... Like, isn't this complaining about like game theory or something like there's not a lot of game theoretic innovations. Like you get like one a century maybe or something.
So I don't think so. I don't think so. I think, first of all, yes, people always say it's a bad system, but it's the best one we have. Mm-hmm. But I don't think necessarily this is the case. I think it's just a relic of what used to happen in the past. It's also quite new. It didn't exist 100 years ago. Mm-hmm. So it's quite new, but with the amount of papers and many things that change over the years. I think bioarchive, the archives, bioarchive is now there's open archive, but it's probably the best thing that happened for us. For biologists, it's only, I think, 11 years or something like that.
It's like the Bitcoin of publishing. You know, it's like the open source version, right? In a way, it doesn't have anything backing it, right? Like, you know, the US dollar has the government, the military, everything, and Bitcoin has nothing, but it has some people with computers, you know, it's like some kind of distributed thing. It's very volatile, you know, there's a lot of similarity.
Yeah, perhaps, but I think it improves our lives significantly. For me now, you know, when I put my paper on bioarchive, then I already feel like, you know, half of the pain is gone. It's out there, people know about it, it gets exposure, it's great.
Right, and legitimately, I think it gets people can use the results at risk already, right? And they can do their work in the context of those results.
Absolutely. In most papers, I read them at that stage anyway. And I take any paper with a grain of salt. I always think that, you know, needed to repeat it and test it and try it and so on.
Yeah, I think probably the biggest problem is that even when the paper is published in Nature, at least when I'm reading a paper that's not in my field and I read a Nature paper, it's like, you know, the word of God. But when I read the paper in my field in Nature, it's full of things that I'm like, well, I don't really, you know, I'm not sure that this is a fair statement. And so in the end, like archive is also like that, right?
I mean, I remember hearing someone give a talk and he said that he started his research by trying to replicate a paper that was published in Nature. And he said it was published in Nature, so I assumed it was wrong, but then it repeated, you know, and then this is what he did. Because of course, the more flashy, nothing, you know, I'm not saying anything about the quality of the review, but the more flashy stuff is published in that paper, in this journal, so it might be less true. But in general, I think, you know, the quality of the review doesn't really correlate with the impact factor of the journal or anything like that. And I think that no, it's not gravity. I think that it's a limitation. The quality of the review is a limit. It has many limitations that have something to do with the availability of the people, with their motives, incentives. But I think that now there's the doomsday machine that could change the way science publication happens and really make a revolution, because otherwise we're just stuck, you know. And of course, this, and I'll explain what I mean by that in a second. I'll just say, you know, the problem is that the paper, where you publish your paper, it will later dictate whether you'll get a job or not, or a promotion, or tenure, and all of that. And the bottleneck is really that the committees, hiring committees, for example, the promotion committees, they just can't read the papers. It's too hard. Papers are too long. The committees don't have the expertise to deal with that, so they have to outsource this to the journals.
So say, what's the impact factor of the journal this was published in?
Yes, something like that, which is totally stupid and everyone knows, but it's the best thing that they have. So what do we do? And the journals don't have enough reviewers. Maybe, you know, Nature can get a reviewer. It also takes them many months. But another journal, which is maybe, you know, a great society journal that's less flashy, but try to do a good job, they can't do it. And this is further complicated now with, you know, open access. Yes, I believe science should be open, but it creates a pressure on the journals, because now they have to publish more papers, because if the papers are open, then they don't get the subscription-based model. They just get paid for the numbers of papers that they publish. So you have more pressure to publish fast, no reviewers, just totally stuck, and they collapse, and then they get bought by the big journals. So the system is really not functioning. It takes months. Now I have a paper under review for 4 months. And it's the usual thing. It's just, and then, you know, who knows what will happen when I get the reviews back. But I think that the doomsday machine, that the thing that can really change this, is the innovation in AI. I think this is, so now, you know, it's only in the beginning, so people are saying, what? People will be using AI to review papers is sort of, it's not the pure way of how we think that science works, but I think that AI can definitely do it. And I think that it's up to us to build this in a way that will be good for the scientists and not for the journals necessarily.
Would you accept the results? Like, let's say your paper is rejected by AI. How would you feel?
So I think if my paper was rejected by AI, I would probably
If it's accepted, I understand.
Right.
So what happens when it's rejected?
And whenever my paper is rejected, I'm always angry. But at least if it's transparent, the system, and if it gave me good reasons and actually, you know, explains all the faults in my paper and blah, 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 at least I know it's unbiased or not biased in the normal ways we think about bias in reviews. So I know that the reviewer didn't hate me. You know, I know that maybe that the review doesn't have a competing paper. I know all of that. Obviously, I think that taking out the emotions and the personal aspect of review would calm me down, actually. Yeah. So, but I think that it's still the way you structure it is crucial. If the journals structure it in a particular way, then it's a problem. But if it's something that is built for the scientists, and is run by the scientists, and I think it could be a real revolution. And this is something that I'm now trying to do now. So it's not published yet, but you know.
Are you actually writing code?
Yeah, so we're
like
doing this. You can even check it out. There's a website. It's not, you know, out in the open. No, so we didn't publish it or anything, or promote it. But it's called QEDscience.com. And if you go there, then you'll see. And this is not something we're giving the journals at the moment. The idea is that actually the scientists would, when they create the work, they would run their paper in the system, get reviews, and improve the work. And it finds problems in your paper. It suggests plans to address them. It's either small changes like just textual edits or experiments that you can do. And you can decide what to do. And it works with drafts. It works with published papers, and also with just results. You can just upload a picture of a gel, and we'll give you reviews, and you can then improve your work. And when you're done with it,
is it focused on bio right now?
At the moment, it's focused on bio. Maybe we'll expand.
One of the kind of like devil's advocate things I can think of immediately is that knowledge. So if you look at all of knowledge, right? Some knowledge is contained in books, other knowledge is in articles or whatever, but some knowledge is contained in people. It's actually not, there's nowhere for the AI to get it, right? So in that sense, like, that's the idea, at least I think, of what the reviewers are supposed to be, is like, you know, they know more than what's in textbooks or in other papers.
No, totally.
So how do you handle that?
So I think that if you give it to the authors, to the scientists creating the work, then they get this feedback and they can respond, and they can, and if there's something that the AI missed, it's fine. It won't be this AI verdict that decides on your paper.
Okay.
But it's something that you can, but it's unbiased, and you get this feedback, and then you can, and this is something, you know, we didn't do yet, but in the future, the way I'm envisioning it is that you get your reviews and you respond. And if you got some stupid comment, then you explain why it's stupid. If it's good, maybe you do the experiment, or maybe you change the text. And there's versioning, so you can see how you improved your paper as a result of the comments that you got. And then it's up to you, just like with bioRxiv, you can post it. So you get your so you don't wait for the journals to use it on you, which will probably, you know, they'll probably build their own tools at one point. So you also want to build this first, I think. But if you're content with the review that you got and you can't mess with it, then you post it, and you have your preprint that you post, and you have your AI review, and then maybe the editor will say, you know, this is pretty impressive. Maybe I don't have to wait until I find 3 reviewers. I can just have 2 reviewers and 1 AI reviewer. Or maybe if there's or if the reviewers don't agree with each other, I can
adjudicate it.
I can check what the AI said, and the AI can also assess how reasonable the ask is, how much time it would take, how much money it would cost, which no human reviewer can do. It can also do other things which practically no reviewer can do, like read the entire paper, or the supplementary, or rerun the analysis. Who does that? So and it doesn't get tired.
I mean, I think you can also, in some cases, people don't want to share all their data, but they can share it with if it's an open source script that's not going to store it, things like that, you can share all the data.
We're not interested in your data. It's just about the comments and the quality of the comments to improve, and the idea is to help the scientists improve their work. You can post it or not, up to you.
Yeah, that's I mean, I think probably secretly this is already happening. So probably a lot of reviewers, when they get a paper, are now uploading it to ChatGPT and just asking, like, what are the problems with this?
Right.
I think that must be happening already.
No, there are a few articles about it, but but
it's not purpose built.
Exactly.
Right? And it doesn't have that I mean, certainly if I was looking at papers on arXiv and it said, okay, this has been reviewed by this known entity, right? That would give me a lot more confidence in.
Right. You want it to be transparent.
Yeah.
To have everything written down and to know that they didn't mess with it.
Yeah, it's really interesting. Can somebody submit their paper now to this? Like,
yes.
It's already available?
I mean, if you go to the website, so QEDscience.com, then you can sign up and you can create the reviews.
Do you have an example on the page? Like, have you submitted one of your papers and kind of like showing what the process looks like? Is it possible to see the comments and things like that?
Yeah, sure. I can show you. Show you later. Yeah, definitely.
Very cool. Okay. I think a lot of people will be interested in that, actually. There's a... Do you think that particular pain point, the publishing pain point, is the core pain points? Like, is that the 80-20 of being, like, so I'm not in academia, so I just kind of spent a little bit of time in academia, and there were these, when I first got to MIT, there were these posters on the wall in the materials department that said like 80% of grad students are depressed. Mm-hmm. If you're depressed, call this number, whatever, right? And I thought, I'm in a very good mood. Like, what's happening here? Like, am I going to be depressed in 2 years? This kind of thing. So do you think the 80-20 is this publishing problem?
I think it's very significant. So there are other problems. Another problem is also, bad mentoring, things take forever, people don't get paid a lot, perhaps. That's also a frustration. But and I think, also the AI can give you review, sort of, comments and help you also with some of the mentoring part. Mm-hmm, so you're not totally, if you get feedback on your work and your plans, so that's also something good. But, and you can do that also with ChatGPT, but again, like you said, it's not purpose built. But I do think, at least for me, that publishing has always been this 80-20 thing. And I think, people don't understand when your paper is rejected for bad reasons. For some people, especially early in their career, it can dictate the entire course of their life. Mm-hmm. You work in biology, you work for 5 years on a paper, and then it's almost random, or worse than random, perhaps, if you consider all these biases, and it really affects your life.
Yeah, I think the results, although biology is like one of the most exciting areas where there's so many results. If you look at the translation to like outcomes, it's not that great, right? Like, I think it's Eroom's law, like instead of it always getting cheaper and better, it's always getting more expensive and worse to make a drug, right? Which is theoretically one of the important things that comes out of biology. So some of that must have to do with the fact that like the whole academic complex is so inefficient.
It takes longer and longer to publish papers. Papers are bigger and bigger. Part of the requirement now is always to tell a whole story from the beginning. That's also,
what's the physiological relevance?
So that's exactly that.
That's one of your big right, hobby horses.
So what's the physiological relevance? So imagine that. Your Watson and Crick, you published the structure of the double helix. What's the physiological relevance? They didn't find, or a mechanism in that paper. What's the mechanism? They also didn't. They certainly didn't find, the therapeutic implications. But now in every paper you have to have all of that. It's ridiculous, and it is because again, I'm not necessarily just blaming the journals or the reviewers. It's just what became standard. There are so many papers that no one reads that you need to select them somehow. So people want the whole thing. And this
it's like a proof of work. It shows that like the idea is so important to you that you spent an extra 2 years on things that probably don't matter that much, but it's like you're so sure of this idea that you're willing to put in the extra work.
Right. And I think that the reason that there are so many mistakes in one of the reasons, there are many reasons. One reason that there are so many mistakes in science is that science is hard. I believe the scientists. I think that sure there are people who go and catch people who cheat. And there's also, softwares for that. But I think I want to believe that's the minority of people who just blatant fraud. On the other hand, I think that the amount of papers that contain some mistake is probably high. And this is because science is difficult. That's especially true in biology, which is just complicated. And many of the weak points in papers, I believe, is just people they had part of the story, and to complete the entire story, they added some part which was weaker, perhaps just to anticipate the ask to tell a complete story.
Have you ever seen this meme? It's like a drawing of a horse, and the legs and the back and the tail are beautiful, and the rest is like a children's sketch.
That's classic. That's classic. Yeah.
Mm-hmm. Okay, so given that you find academia so painful, and to the point of generating all this humor,
but
right?
I also think it's great. Okay.
Of course. So that's my question. What makes you excited about it, and what in your research like keeps you going? Like why do you love it?
So I love it for many years. I think it's the best job in the world. I think it's hard to get to a point where it's where you're totally free to do whatever you want. So it's a very long process and so on. But I love I think it's a loophole in the world to be a scientist. We don't go to work at the same hours like normal people. We do whatever we want. We have a complete control over our schedule. And a choice of what we do. It doesn't have to bring profit in the end. It doesn't have to be useful in any way. It's great. This is all amazing. We get to as PIs, as professors, we always work with young people. We work with extremely smart people all the time. New influx
at the peak of their cognitive abilities.
Yeah, at the peak of their cognitive. And so all of that I really love, and the freedom. And for me, the best thing is that I can switch between different topics, and we'll talk about, I hope, about different things that we do. So all of this I really love. I think that there are many challenges. Another thing is that the challenges keep changing. Now, when you just start, you're an undergrad. Who remembers? I don't even remember how it is. Just when I was, getting coffee this morning, I heard some undergrads talk about what do they need to do to get the credit for the project or something. Who cares? All of these things or exams. It seems like in another world, totally unrelated to my life. And it's also true for different stages. So you have challenges finishing your PhD, and then you have challenges when you're a postdoc getting a job, and then getting promotion. So it keeps changing. The problem for me now is, what do you do after you have tenure and you have the promotions and you're there already? And because, in every stage you need to do something to pass to the next stage. But then when you're 40 something, now you need to you need to work on that for 30 years. And then the question is, how do you keep doing this and maintain the interest and the enthusiasm? Because, papers you publish papers. Most of the time, it doesn't translate to some major change in, to a drug or to a huge discovery, because science is accumulative. It happens in small steps most of the time, and also there's a lot of luck involved. So how do you maintain the enthusiasm? I don't know the answer, but what I do know is that waiting 5 years to publish a paper is not the way, and then getting ejected and then spending half of your energy just on revisions and resubmissions, that's a grind. And I want to cut that grind. That's
I think that's probably shared by a lot of people, that feeling. I guess it's a good segue to ask you about what it is that's happening here. So we're in your lab. There's students, going back and forth, manipulating something under stereoscopes. What's happening here?
Many things are happening. Most of the science that we do is conducted with C. elegans nematodes, which are very simple organisms for one of the main model organisms for studying biology. And we're using them, I would say, about 50% of the people in the lab are using C. elegans, but we're also studying many other organisms. And the main topic that we have been studying is epigenetic inheritance. Epigenetic inheritance is inheritance that occurs independently of changes in the DNA sequence. But, and I'll be happy to talk about this. I can talk about it forever. But we are also studying many additional non-related topics, because as I said, to keep interested, I have to try other things. So we've been studying brain parasites, engineered brain parasites, engineered to deliver drugs to the brain. We studied economic irrationality. We studied the Dead Sea Scrolls in Qumran. We are working on many different things.
So let me ask you, before we get into the heavier, science questions, I went through the Dead Sea Scrolls paper a little bit. So from what I could understand, basically the Dead Sea Scrolls are written on animal skins, and people had already figured out that you could amplify the DNA in these 4,000, 3,000-year-old skins, and maybe sequence parts of it or something like that. But they're all fragmented, right? So they're broken up. And you guys did you do your own sequencing and amplification, or did you just take the data and process it, or like what was your
No, in the past, so in the past only people amplified them with PCR and did some Sanger sequencing, not deep sequencing with sequencers.
So you were the first to basically actually use modern methods like Illumina.
Exactly. And also now the standards of ancient DNA are very different. These are experiments that were conducted in the 90s. So now when you do these type of things, you work in clean rooms.
So it's full of human DNA, also modern human DNA.
I mean, also our sequencing is full of the human DNA. We took all the precautions, to get rid of it or to filter it out later bioinformatically. But there are contaminations there that are also ancient. Probably, we didn't find any ancient DNA of humans. And this you look for degradation patterns that characterize ancient DNA. So we didn't find ancient human DNA. We did find ancient animal DNA, and we sequence the different animals, and what we try to do, the different pieces, so there are 25,000 fragments of skulls, and the challenge is trying to assemble them, the
So
different
just
pieces.
for people that don't know much about this, and honestly, I didn't know much about this, there are caves next to the Dead Sea where it's extremely dry, and so you have these preservation of these scrolls that were left there for one reason or another, and they're in Hebrew, so you can read them,
Right.
basically.
Right. So this is considered to be the Dead Sea Scrolls were found in the 40s and 50s in the in Qumran, which is a place near the Dead Sea, and also some other places in the area. And these are considered to be this is considered to be the most important archaeological finding we have in Israel. And we have lots of archaeological findings here. And one of the
It's a
reasons...
military base and archeological site. Those are the two major activities,
There are all kinds-
activities in Israel.
Yeah, probably in cyber now, so we're strong at cyber. It's the oldest Bible. So the next one is 800 years older. But it's not only the Bible. These are many texts that were found there that are very important because they were written around the year 2000. So this is a very crucial time for both Judaism and Christianity in Israel. And most of what we know about this area, about this period, is coming from most of the texts what preserved was from the Dead Sea Scrolls. So they found 25,000 pieces of fragments, and the challenge is trying to assemble them. And people have been assembling them for many years using different techniques, like putting a puzzle, trying to connect the pieces that are connected, or using their color or shape, ink, all kinds of clues. We used high throughput sequencing to try to say this piece and this piece came from one animal, this piece came from another animal, so they go together. And we did this, and this was a super cool paper that's totally out of the normal stuff that we do in the lab.
What was the hardest part technically? Like, why was this a technical challenge at the time?
First of all, when we started doing this, we didn't know what we were doing. So we had to collaborate with experts in ancient DNA, Matthias Jacobsson's lab in Sweden, in Uppsala.
And I guess they're sequencing like Neanderthals or something.
They are mostly interested in ancient humans. Yeah. And my lab manager, Sarit, traveled there, spent months in Uppsala. She's my, for special tasks. She's there. Now she's in Cold Spring Harbor in New York, sequencing something else, which is also using a new sequencing technique. Not ancient DNA, just some new sequencing technique. But so she spent months in Uppsala, and we had to sequence extremely deeply, billions of sequences, to get the coverage that would allow us to actually do some piecing apart. It's very challenging because most of the DNA is in very low quality. And we also just can't take an ancient piece of these super precious scrolls and grind it.
It's like dust and crumbs, basically.
Exactly.
That
This is
you were
what
allowed to use.
dust and crumbs, and also some when in the I don't know. This was probably in the 50s, or I don't know exactly. But when they started assembling the different pieces, the way they did it, they just took big tables and they put the pieces. And when they found a piece that matched, they glued it together with duct tape. Okay. They didn't think about the consequences back then. And then for 30 years or 40 years, they've been removing the duct tape.
Oh my God.
So we started by, they also used hot glue, and then it's, dead. But we started, but when they remove the duct tape, they keep the duct tape next to the piece. So in the beginning, just as a sort of a proof of principle, we sequenced from the duct tape. So we also had to use, tiny amounts of materials. So it's very technically challenging. It wasn't clear at all that it would work.
How long did it take?
It took 7 or 8 years.
Wow. So you have to have some stamina to...
Yeah, you have to have stamina. In this case...
I assume that this wasn't funded like this, or did you have
was
funding
funded
for this?
in the middle.
Okay.
I'll tell you about this in a second, just about the stamina. Well, again, science is connected to the structure of science. So you can't have a PhD student, for example, do all the work because they don't stay for 8 years. So this was led by Sarit, who's my lab manager. And she opened the lab with me 13 years ago when we just started. So she's still here. We did get funding in the middle. So we started just with my startup money when I opened the lab. But then it was funded by Eric Schmidt from
Google.
From Google, you're right, who was the CEO of Google. So he visited Israel, and the university arranged for a meeting with him and Stu Feldman, who is the head of his science endeavors. And I told them about this, and Schmidt said, this is the best idea I ever heard. And two weeks later, I had the funding for the project. But that was very fortunate. It's not always like that. I have many crazy ideas, not always have the funding to do them.
Yeah, that's awesome. What was, in terms of the outcome, so you were able to basically say more definitively these two pieces or these n number of pieces go together.
Right.
Were there any new like historical insights that came out of that?
Yeah, so one thing, I mean, you told me before we set up that you're going to Masada, right, for a trip now. So one of the questions, there were some questions about particular piecing together and apart of specific fragments, but there were also questions about the collection as a whole. So for example, I mean, as I told you, most of what was found, what is in Qumran, which is a particular set of caves there. This is also the first cave that was found with scrolls is in Qumran. And then the question is, imagine that Israel is bombed with a nuclear weapon and everything is destroyed except for the library of some guy living in a cave in the desert, in the hottest place. You'll see when you'll be in Masada, maybe you've been there before, it's super hot. It's hard to live there.
So for people listening that don't know what Masada is, it's a fortress that the Jews, during the Roman period, some number of Jews went up into this fortress that was supposed to be impossible to, it was like very defensible. And they stayed there for a long time, eating pigeons and so on, until the Romans finally built a ramp up to Masada, got in there, and they all ended up committing suicide, except for one guy. The last guy, they were all stabbing each other to commit suicide. The last guy, Flavi, Joseph Flavi, is that correct?
Josephus Flavius.
Josephus Flavius ended up getting captured by the Romans, I think because he just didn't kill himself or something like that.
I don't remember exactly. I'm not sure that it was him and he didn't hear about it from someone else. I don't remember the story.
I thought he was the one survivor or something.
I'm not sure.
I could be wrong. This could be, it should be fact checked. But basically he ends up in the Roman Empire, I think in Rome, and he ends up writing the history of this. And that's how we know something about it.
Yeah, but all of this area, including Qumran, which is a different place, is extremely hot. It's really hard to live there. And imagine that the entire country is wiped out. And all you have left is by chance, the library of someone living in a cave in the desert. Okay? The question is, how representative is his library of what was going on in Tel Aviv, for example? Okay? It's very different. You have to be a special guy to live there. And most of what was found in the caves. So we learned a lot about the history, but is it representative of what happened in Israel in general? So most of the scrolls were found in Qumran, but there were also scrolls that we sampled and sequenced found elsewhere, like in Masada. Masada was the last stronghold. And some scrolls were found there. So the question is, did the people in Masada, the scrolls there that were kept by these people, did they bring them with them when they escaped Qumran? Or did they have their own scrolls with the text? And some of the texts that were found are very characteristics of Qumran. For example, there's a song that it's one of the oldest ancient mythical songs. It's called Songs of the Sabbath Sacrifice. It's classic Qumran. And it was also found in Masada. The question is, was it brought from Qumran or was it known throughout the entire country? And what we found is that the sheep that were used to make the scroll in Masada are totally different from the different breed of sheep. Right. Different breed of sheep than the one found in Masada, which suggests that perhaps it was more common throughout the land. So this is sort of like a meta thing that we learned.
That's very cool. I think it's very reminiscent of there's this whole thing going on. There was a scroll prize for decoding the Vesuvius scroll. So there's all these completely burnt scrolls from an enormous library at near Mount Vesuvius that people are now decoding with AI. So they're X-raying them and decoding them, and maybe we're going to get like double the amount of Roman texts or something like this. There's a lot of excitement about this, kind of similar but bio version of this.
Very cool. But as I said, this is really not my day job. So my day job is mostly focused on epigenetic inheritance. But that was a fun project that emerged, which I enjoyed a lot.
So I have a lot of questions about your epigenetics work. I think maybe a good place to start is basically, I think the question that's interesting maybe to lay people is whether, there's the idea of like gene editing humans, like gene editing embryos, for example. There's another guy that has a very popular Twitter account. I never can pronounce his name, but John Kuhei, the CRISPR baby Chinese
That's
scientist.
extremely controversial, of
Yes,
course.
and
yeah.
he's been posting these incredible things on Twitter where he takes a photograph of himself in the lab and then writes a very controversial statement.
I saw that.
He invented a new format. This is like a new format of meme that's just him, saying like, gene editing will cure all disease or whatever. Like he makes some very controversial statement and then posts very like the pictures that he takes look like Instagram model type photos, but it's him in the lab, Wow.
Of course. I mean, he when he just announced it, he disappeared for a while, and it wasn't clear whether what it is ethical and so on. Yeah.
But basically, I think the question, like a most like high level question you can ask is, if we accept that epigenetics has a significant effect on humans, not just on and of course, like your research is, you're one of the people showing that there's this huge effect in animals. If we see that there's an effect in humans, how realistic is it to do like epigenetic editing of some type?
Right. So first, what we showed in nematodes, C. elegans nematodes. There we understand epigenetic inheritance, transgenerational epigenetic inheritance quite well. We know the mechanism, and the mechanism there is inheritance of small RNA molecules which regulate gene activity. And we know about genes that function in small RNA inheritance. We know how the RNA moves between cells, how it gets to the germline, gets amplified. We know a lot about it, and many people are studying it. No, close to nothing in humans. Okay. In other organisms, it's still controversial. In humans, I think in nematodes, part of the frustration that it's not controversial anymore. Okay, it used to be very controversial, and also for that reason, probably easier to publish. Now it's people believe it. It's a problem because you need to step up the game somehow. But in humans, we don't know anything pretty much. I'll add that we are also missing many details in nematodes. So what I say, there are still reasons to publish this in nematodes. We have a lot to learn. But in humans, we don't know anything. A few things about that. First of all, 4 out of 5 animals, just numerically, is a worm. Okay, so worms are very important for understanding biology and life on Earth. Second, I think that if this will be found to be conserved in humans, and it's a big if, we don't know, time will tell, it's much harder to study epigenetic inheritance in humans, then I definitely think that it could be applied. I think that before you go and edit the epigenetic information, which is something you might be able to do, the first thing that you could do, and again, there are ethical problems with that, and there's a very long history. It goes, well before the person you mentioned to the ideas of eugenics and improving people. So this is eugenics started with Francis Galton before or you can even go to the Greeks if you want, but Galton talked about it the way we know about, before they knew anything about DNA or just how genetics or Mendel's work and so on. They didn't know anything. Still, so the problem is much deeper. And epigenetic is just another layer of that. I think you can edit the epigenome and change it in many ways. There are already the tools to do it, but you can also select based on epigenetics. For example, you can characterize the pool of heritable small RNAs in sperm, and this would be correlated with certain traits, I imagine.
And that's not shown in humans, but based on the work you do and the work that other people do, it's plausible that it might be the case.
It's plausible. So some of the machinery is not identical, and you can argue this way or that way. So in C. elegans,
the biggest difference, I guess, is that there's no mechanism to amplify the small RNAs in humans.
Right. So
there's no known mechanism.
There's no known. In C. elegans, there's a trick. They have these enzymes that are called RNA-dependent RNA polymerase, which can amplify and generate more and more small RNAs in every generation, so they don't dilute. C. elegans are not the only organisms.
To be clear, you inherit. So you start the animal with one cell, and it has these small RNAs from the parent. But then if you divide that cell many times, it's going to dilute out the RNA.
Exactly.
So you need a way to amplify it if you want it inside every cell.
True. You need to amplify it so it won't dilute over generations. And in humans, we don't have the same machinery that we do in
Are we 100% sure that there's no amplification in humans, or we just haven't found it?
We are not 100% sure. Okay? Before I can say something a little bit more technical about that in a second, but the machinery exists not only in C. elegans, but also in other organisms. Okay? In humans, we don't have the same enzymes that are called RNA-dependent RNA polymerase that the worm has, but there could be other ways to amplify the information. For example, there are very simple viruses that amplify, RNA viruses that can amplify, even though they don't have RNA-dependent RNA polymerases, like hepatitis delta virus. In the past, it was thought that the amplification occurs by another virus that brings the machinery and helps the other virus to replicate. Now people know that this is not the case. And there's some endogenous factor that helps the virus replicate. It could be just the DNA-based polymerases that
so you first convert the RNAs to DNA, amplify them, and then back to RNA.
No, but some DNA-based RNA polymerases have also the capacity perhaps to amplify RNA in certain situations. So that is one possibility. The other, which is not proven, but it's one possibility. The other possibility is that the information will be that there will be a few different epigenetic processes occurring. So, for example, in plants, small RNAs can also amplify. There are RNA-dependent RNA polymerases in plants, but in addition, small RNAs can also guide the deposition of different chromatin modifications. So the information can start as RNA, but then it can be translated or changed into an information that is held in the form of some sort of modification, either of the DNA, like methylation, or of the histones, which condense the DNA. Is that
conserved across replications of the cell? So like do those histone modifications get conserved as the cell divides?
So that's another big question that many people are studying. What type of chromatin modifications can be preserved across cell divisions? In plants, DNA methylation of certain type of DNA methylation can do it. And there are also proofs that in some organisms, and depending on the chromatin, on the histone modifications, that some modifications that can also survive DNA duplication and cell division. But we still have a lot to understand there. And then the information which is held in histones or in chromatin modifications can also be translated back to the production of more RNAs. So all of these feedback loops exist. To what degree do they exist in humans? It's unclear.
So I have two questions. One is, first of all, what is in C. elegans, what is the most salient proof for this small RNA idea? Right. So what's like the strongest proof that you guys have found or other people have found? And my second question, which is a follow-up, is just, are you tempted to start working on human cells?
Yeah. So you ask a lot. I'll explain. I'll try to explain it relatively briefly. Okay. And that's another advantage. So let's start with the advantages of C. elegans for doing this type of research. So C. elegans, aside, you mentioned that they have a very small number of cells, just 959 cells, 302 neurons. That's one advantage for many things that you want to study with them. For transgenerational inheritance and epigenetics, there are two other advantages, or three other advantages. One is that the generation time is very short. It's just 3 days. So you start an experiment with one worm, and then after 3 days, you have the next generation. You want to do this in mice, it will take you months. In humans, it will take you 20 years. Okay. So that's one advantage. The second advantage is that you can really separate nature from nurture. So you want to control. You want to know that the effect occurs if you change something in the environment, and you want to see whether it infects the next generations. You want to be sure that you can control their environment very accurately. And worms just live on petri dishes in the lab, and they eat E. coli bacteria. And you can control the temperature that they are growing in, and they're almost. So if you start with a single worm, then it can self-fertilize. It's a hermaphrodite. So it makes descendants that are almost genetically identical. So you also can rule out DNA differences as the source of the effect. In addition, so this is one reason why studies in C. elegans of transgenerational epigenetic inheritance are so strong. In addition, there's one more advantage, which is really a game changer, which is you can just feed the worms on RNA, on bacteria that produce RNA, and they will eat this RNA, and this RNA will now start a silencing response or gene regulation effect.
When I was preparing for this podcast, this is the thing that I understood the least. Why don't they have a way of, doesn't it seems dangerous for them to just take any RNA that's in the environment? Why don't they have a way of excreting it or destroying it or something like that? So the ecological relevance or how it is, it's just not unknown. It's totally unknown. So it's a very good question, which is still unanswered. But I have to explain perhaps what is RNAi, because I know non-biologists are also listening.
And this was discovered in plants and in C. elegans. The Nobel Prize went for the C. elegans people in 2006. Another Nobel Prize for RNAi really was awarded this year for Ruvkon and Ambrose. The first Nobel Prize for Fire and Mello in 2006. But when you feed worms with double-strand RNA, we all know that DNA is double-stranded. But RNA can also be double-stranded. When you feed the worms with double-strand RNA, when you inject them with double-strand RNA, when you soak them with double-strand RNA, they take it up into their cells. Mm-hmm. And this double-strand RNA is something that perhaps is conceived for the cell as something foreign. So it's being chopped into small RNA molecules, which are just literally short, 20-something nucleotides long. And these small RNA molecules can then find genes, or more accurately, mRNAs, messenger RNA of genes, and by base pairing with these mRNAs, lead to the degradation of the RNA, mm-hmm. or prevent even the transcription of these RNAs. So these RNAs have a role in regulating which genes will be transcribed.
Is the reason that they get degraded that suddenly it's a double-stranded RNA and it's not, so it's foreign or something's wrong with it and there's an enzyme that chops it up?
There are many mechanisms.
Okay.
It's quite complicated. There are also different ways that they do it. But the bottom line is it's a way for the cell to recognize genes that need to be shut down. Mm-hmm. This shutting down is called silencing or RNA interference. Mm-hmm. Okay, so when you and it's surprising, like you said, that the worms can just take it up. You can just soak them in double-strand RNA, they take it up. It's possible that they do it to communicate with other worms. It's possible that they do it to take RNA from the outside as food. It's possible that they are being manipulated by some pathogen. This is just not known. Mm-hmm. And many people try to explain this, and it's still poorly understood. And indeed, one of the C. elegans has so many advantages, but we don't know enough about its ecology and like the natural context. And this is something that people are now starting to look at. Mm-hmm. Okay. But the bottom line is you can use it as a tool and to study genes and gene regulation. So you can just, and you ask, what is the most salient proof that transgenerational epigenetic inheritance occurs in C. elegans? Because we didn't emphasize it enough, but transgenerational epigenetic inheritance, or the idea that the environment can change the next generation, is a taboo in biology. Mm-hmm. Perhaps the biggest taboo in biology.
Lamarck, Lysenko. It's the biggest taboo in biology. It's ancient, goes back hundreds of years, at least, depending on when you start counting. And it's the one red flag with the most, it's like, it's the thing that annoys biologists most. So this is not something, just some small thing. Yeah. It has a very, very long history. I teach a course about it. I can talk about it forever. But for someone who's not familiar, this is like the biggest taboo in biology, probably. It's like telepathy almost, to say that, that environment or that life history can lead to transgenerational changes. But in C. elegans, it's now proof. And you ask, what is the most salient proof that no one can argue with? And this is that you can just take worms and expose them to double-strand RNA. Mm-hmm. And when you do this, you feed them or you inject them with double-strand RNA that corresponds, so it matches in sequence a particular gene, mm-hmm. the mRNA of a particular gene that has a phenotype. So if you feed them on this RNA, double-strand RNA, the RNA will be chopped into small RNAs. It will lead to the silencing of the mRNA, of the corresponding mRNA, just this mRNA, not other mRNAs. And this would lead to the silencing of a gene. It won't be expressed, and there will be a phenotype if you're silencing a gene that has a phenotype. So for example, you can silence a gene, and then the worms will start moving weirdly. Or maybe they will just become very short. Or maybe they will die. A very short, or very strong phenotypes. You can also just silence a fluorescent protein, then they won't be green. Normally mm-hmm. they're shining green, now they won't be green. And then what you do is you stop feeding them with this, you stop giving them this RNA, and you can see, and this was done by at least hundreds of labs, maybe more, you can see that the effect continues for the next generations. Mm-hmm. So they are not exposed to the RNA anymore, but still the next generations are affected for multiple generations. It's not just diluting of the RNA that you gave them. It lasts, the longest effects that have been documented last for 5 years or hundreds of worm generations, wow. long after the original RNA is gone. In every generation, you'll have 300 worms. So it mm-hmm. will be diluted very quickly. After 4 generations, it's dilution by billions. Okay? And so this is very clear cut. You know exactly what you're looking at. You're silencing a particular gene. You have a control. We talk about, the basic things you need to do. You can silence another gene as a control and it doesn't lead to the same effect.
And so this is something no one can argue with. And not only that, by now we also know a lot about the mechanism. So we know this doesn't happen in certain mutants that are defective in genes that code for the machinery that amplifies the small RNAs, that shuttles the RNA between cells, or that have to do, genes that have to do with producing of new small RNAs, or by affecting the chromatin, because these small RNAs also affect the chromatin and function in the nucleus.
Is it so that's a nice controlled example, but obviously like in nature, they're not being exposed to RNAs in nature, or not obviously, like not in normal situations. Some of the other examples that, I read about were like if you starve them in the next generation, they're going to have some, some response to that, right? Like some change their metabolism or whatever. Is there proof that's as strong that that's being inherited through the same mechanism that's easy to study by soaking them in RNA?
Right. So different environmental conditions, certainly not all environmental conditions, but some conditions such as starvation and temperature also lead to transgenerational changes. And there you look at the phenotypes. You can also sequence the small RNAs that are inherited and look at gene expression, mRNA data, and you see the changes. You can do control experiments which are comparing starved worms of descendants of starved individuals or worms that were well-fed. And when you look at phenotypes, it's harder because many things change. But still, you can do the control experiments and you look at thousands of worms and you compare the inheritance in mutants that are defective in RNA inheritance, small RNA inheritance. And you can prove it quite convincingly.
So the effect goes away if they don't have this mechanism of amplifying the RNA.
It goes away if they don't have the mechanism, and it also goes away after a few generations, so it doesn't last forever. And now there are also reporters. For example, there are genes that are expressed just in high temperatures. Okay? And then you can have a fluorescent reporter reporting this gene. You see that if the ancestors grew in hot conditions or normal conditions, it leads to transgenerational changes in the levels of these reporters. So in addition, looking at complex phenotypes, you can also look just at genes that report the effect.
If you were going to do like a experiment in humans or in human cells or something like that, that would give this kind of evidence, like this level of evidence, what would that experiment look like? So is it possible? So
it's great. So you asked me whether I'm tempted to study human cells or other organisms. So yes, I'm definitely, not only am I tempted, I'm actually doing it, studying many different organisms, including human cells. And there, the key for me is trying to have organisms or tissues that I can expose to RNA and then look at just the silencing of a particular gene. Because that's much more powerful. It's just an easier lens to look at. For example, even if you talk about normal genetics and you want to study complex traits, like height or intelligence.
There's too many genes.
Many genes change, and each has a very small contribution, so it's harder. Statistically, it's just harder to do the experiment. If you start by silencing one gene, then you have a much better handle. So when I go to study this in other organisms, I try to look at organisms that you can start an RNA response just by and silence one gene just by exposing the animal to double-strand RNA.
But this is going from the bottom up of saying, is this RNA effect, does it exist in this animal? I guess the top-down approach would be to say, okay, we have this thing that we think is epigenetics in humans. Let's say, there's some responses to starvation also, right, that are well-known. And can we find the RNA mechanism for this?
Right.
Like, what would that look like?
So I tried to do things like that a while ago, and I sort of gave up. I think it's very hard.
What's the challenge?
So in humans, if you want to do something like that, I never did. The challenge is, you have to control for so many things. I'm not even sure how to design such an experiment. And if we mention these experiments, regular genetics, when it comes to complex traits. So for these experiments to become convincing, like
you need tons of people.
Right, genome-wide association studies, they start working only when you look at hundreds of thousands of people or things like huge populations. And imagine you try to do this for epigenetics. Now you don't need just their genome. You have to control for the environments, and all of this is it's really hard. So maybe this is a challenge for a sperm bank or something like that, right? Which I don't have. But
So part of it is just the N. You need a large N to kind of smooth over all the variation.
right. So this is something that maybe a company like MyHeritage could do or something. But
Like
it's very
do RNA sequencing on a massive pool of human samples.
yeah.
What's the N? What do you think is the N?
I don't know.
Okay.
I don't know, but I imagine it must be very high.
So the experiment would look something like this. It's like, okay, you have some group of people that was exposed to some event that we think, based on other considerations, people think has epigenetic effects. And then you sequence their RNA, small RNAs.
And to make things easier, I would certainly focus just on RNA from sperm or something like that.
Okay. Oh yeah, because that's what's passed on.
Exactly.
Right? So you sequence the sperm RNA, and then you sequence, control healthy population, and then hopefully at some large N, some effect emerges. So you can see that there's a contour of an effect that
Right.
might be related to the phenotype things
Right.
that are being noticed.
Yeah, but to complicate things further, this might work, okay? But it will only be what we call an intergenerational effect, okay? To separate from transgenerational inheritance, which is what we study in C. elegans. Intergenerational effects are effects that could result from exposure of the parents,
Mm-hmm.
of the germ cells in the parents.
While they're actually in the parents.
Yeah.
I
so,
see.
so for example, you mentioned starvation experiments in humans.
Mm-hmm.
The famous one is the Dutch famine, but there are other big starvation experiments. In the Dutch famine, which is people were starved during the Second World War, the end of the war, and then the descendants were shown to suffer from all kinds of things. And also later, the grandchildren.
Mm-hmm.
Okay? But in these experiments...
Yeah, I'm basically one of these people, but not for of course, the Russian,
okay.
the World War II
Right.
in
There
Russia.
are also
But same war,
there are also
same
starvation
situation.
in Russia and in different places, China. But in these cases, in the Dutch famine example, the people that were compared were exposed in utero.
They were conceived during the famine.
Right. And they were compared to other siblings that were not.
Mm-hmm.
And so obviously, while in utero, if the mother is starved, then the children will be affected. Not only that.
They're directly getting the information. It's not coming from the mother
you don't
per
need
se.
to come up with a new mechanism of
Right.
inheritance, which is not. Not only that, when they were in utero, they already had their germ cells. So even the grandchildren were directly affected.
Yes.
Only when you go to down the road to other generations that were not exposed at all, you have to evoke some other mechanism of inheritance. So if you do this experiment that we mentioned about sampling the sperm, it will be interesting, but it won't be that.
Is there an example of human epigenetic inheritance that's more like clearly separated, like where it's not while the baby's in the womb?
Yes, so there are experiments like that, but they're all phenotypic.
Mm-hmm.
They're not at and then so people claim there are different publications going further down the road. Or if you go through male, you don't have to go for so many generations. If you go to the F2, that's far enough. But then the question is, what's the mechanism?
Mm-hmm.
And when it's such an important and controversial question, you have to, which is because the reason that it's not trivial is that we don't know of this of the small RNA mechanism that is described in worms. We don't know that it exists in humans. And the other mechanisms like DNA modifications and histone modifications, there's in the germ cells and in the early embryo in humans and also in mice, there are two waves of reprogramming where most of the marks are removed. So it's sort of a reset. Okay?
Before in the embryo gets reset.
The embryo and the germ cells.
Mm-hmm.
And so you have to explain how the effect goes if there's this reprogramming. It could be RNA, or it could be that the reprogramming is not complete. We know that the reprogramming is not complete.
Mm-hmm. So
So depending on who you ask, some people will tell you, 90% reprogramming is severe, is a lot. But some people would say 10% is, it's a lot.
10% is a lot.
Depending who you ask, okay?
Yeah.
But then you have to, but this is the challenge. And so this is why it begs the question, what is the mechanism? And you need to prove it perhaps first in germ cells.
Mm-hmm.
Do you have an amplification mechanism in germ cells?
And what if you allow yourself to kind of dream, what's the biggest impact thing that can come out of it? Let's say we figure out the epigenetic inheritance mechanism in humans, and we can start doing something to it. We can start affecting it with anything from like lifestyle to drugs to whatever.
Right.
What is the biggest problem we can solve, or what's the biggest opportunity we can take advantage of? Like, what would this give us?
The opportunity is that unlike DNA, the DNA it doesn't matter what you do, it stays the same. It doesn't change in response to the environment. But imagine you have a particular epigenetic pattern that correlates with a healthier lifestyle. This might change, this might be flexible. Maybe people will be told, before you go have your kids run on a treadmill or something, or eat a particular diet, because this will change your epigenome, which is plastic, unlike DNA.
And but let's say we figure that out and we have people eating the ideal diet, right, or whatever. What's going to change? Like, is it small changes? Like, what's the big? Can we, for example, with DNA reprogramming, you can like make people. I mean, the famous examples, you can make people. If of course they're probably off-target effects, but you can make people resistant to HIV, right, which is a very big deal. What's like the most impressive thing that can be done with epigenetics potentially?
So I to be honest, I don't know, but there's a lot of discussion, which is by itself controversial, about missing heredity. Some diseases, for example, are clearly heritable based on twin studies, but the genetic basis is not found.
Unknown.
So perhaps, if that is at the layer of the epigenetics, then maybe you can correct these diseases. Schizophrenia, diabetes, things like that.
Okay, yeah, that's really interesting.
But of course, I don't know this is the case. It's just speculating. Yeah.
How much do you think about the overall, I think there's this like a very dark matter region in biology of like you have the genetics, which can be studied, or, RNA, DNA, etc. And then you have the outcome, which is this animal, right? How much do you think about like the, what's fundamentally going on in between, right? Like, what is this like I don't know, I almost think of it like a neural network or something that converts this data, whatever the data is in the embryo and between the proteome, the RNA, the DNA, everything, into the outcome. Like, do you think about this a lot? Do you think there are like some of your research can be applied to like learn more about that, like just bridge that gap?
Yeah, so developmental biology, what you describe, is a huge field. So much things going on. It's extremely complicated, but people are figuring out a lot of the steps for generating an animal. It's hard to do in C. elegans, with its 959 cells. Of course, it's much harder in more complicated animals, but it's fascinating and it's amazing.
Are we even aware of the mechanism that prevents 960 cells from forming? Like, what's killing that 960th cell?
Yeah, so we know a lot about that, in fact. And, so apoptosis, and this is the sort of things that have been found studying C. elegans. And the entire lineage.
But where's the clock? Who's counting to 960?
So a lot is known about it. So we start, we know about the entire lineage from one cell till you get to 259 cells. We know about the origin of every cell and from which cell it was derived, and what is the program for generating many of these cells at the end, and also how to prevent them from further dividing. And
is there like a chemical computer that's basically keeping track of the number of cells, or is it like a local thing that's happening in small patches of cells? So is it like many independently controlled systems that add up to 960, 959? Like, what's the
I think about it
loop?
like that. So in each cell you have this computation of whether to continue, of whether to differentiate, and so on. And there are also gradients that form, larger scale patterns. Yeah.
But it's amazing that it's controlled to that level, like numerically, right? And that is there like a specific protein that's known for or it's just like a compendium of different factors that end up controlling it so tightly, or like what's the?
So it's many different things. But it's amazing that the number is so precisely the same, and also quite conserved in many different nematodes. That for a number of neurons, for example.
Oh, really? So it's not just C. elegans. This is a mechanism that exists across.
Right.
Wow. Yeah, that's incredible. Okay, I have a related question. So, what makes a good model animal? And what's missing? Like, do we need another model animal? Like, I have a friend who, when I lived in Israel, was doing research at the Ocean Institute, and they have these Bacillus. It's like a animal that grows in ocean water. That's it has a lot of strange properties that I think they're always pitching it as a model organism because it's like it consumes its children to like replenish itself. It's immortal because it consumes its own children and like somehow becomes immortal this way. So what's missing in model animals?
So great question. So I think that, first of all, if I had $1 billion to build an institute in biology, I would definitely build it for an institute for new model organisms. I think you get the most bang for your buck when you start a model organism. You see, if you look at all the main model organisms, how much has been discovered. C. elegans, Drosophila, Arabidopsis, zebrafish, all of them. Amazing number of discoveries just because people decided to focus on one animal and study it. And different animals, they show you different things. Now, and some things you'll only find if you focus on one particular animal. And sometimes, and if you choose the wrong model organism, you won't find. For example, C. elegans, RNAi, or everything that we discussed. If people studied not C. elegans, but C. briggsii, which is another closely related nematode, then it doesn't take up RNA from the environment. Okay. You can make it do it, but maybe RNA would never be discovered, for example. Okay. Now, I think what makes a model organism? First, you need to be able to do certain things, to have tools to do certain manipulations. You need to be able to grow it and cultivate it. But eventually, now there are many animals or organisms that people study that are non-conventional model organisms. At the end, whether it's a model organism or not is decided by whether there's a big community of researchers that decided to study it. And if there's a big community that create all the reagents and all the tools and collect the information and share it, then it's a model organism. That's the only definition. Okay? Now there are people trying to make spiders into a model organism. It's amazing how they build a web and do the computation to do this. So many interesting model organisms for so many questions, but you need to have a community formed. And I can think of a million different model organisms.
Are there funding mechanisms to generate model organisms?
A little bit, but not so much.
Okay, yeah, that's interesting. And you think that's one of the highest leverage, that's actually one of the highest leverage things in biology. Basically generating a model finding a model organism that helps explore a difficult question.
Look at the amazing success of the different, in C. elegans, 8 Nobel Prizes since the year 2000.
Wow.
So it's really, I think it's that's certainly the way to go.
How long did it take for C. elegans to become a well-known model organism?
So it started abruptly because Sidney Brenner in the 70s wrote a grant, which is like half a page, for starting his own group around C. He was already a famous scientist for studying phages and the DNA, the code, and he discovered so many things. And then he said, I want to form a group around C. elegans. And it started by, I don't remember the exact numbers, but maybe 30 people or something, which, and they, it goes back to the beginning of our conversation. They didn't publish for 7 or 8 years. They didn't publish a single paper because they were just creating everything that they need. And then they made a revolution and so many Nobel Prizes.
What was their first result?
So they published the mind of the worm, which is the connectome and all of that. And,
wow.
And they worked for many years on that.
Was it easier? Like, do they have large neurons, or there's clear, right? C. elegans is see-through. That's part of the benefit.
It is see-through, and so for the nervous system, yeah. So it's see-through, and it's in the development of the nervous system is totally stereotypic.
Right. Yeah, which makes it much easier to study.
Right, but they had to write the drivers for the printer and all of that, back then, and they did it. Sidney Brenner did, I think, the driver for the printer. But
what do you mean by the driver for the printer?
It was before he had to write the program to print or something.
You mean like to print? Explain. I don't know.
To print pages with the pictures of the
I see.
of the neurons of the EM or something like that. I don't know exactly, but they had to do everything, and it's amazing that they had an EM reconstruction of the entire nervous system.
Wow. Okay, that's interesting. So basically, the first hook with C. elegans was this. The nervous system is very predictable, very easy to study.
That was the motivation of Sidney Brenner to study the worm, and he debated between a few different organisms, including also tardigrades, these water bears, which was another organism he thought about, which are much more complicated. They have thousands of cells,
Water bears are larger than
No,
C.
they're
elegans?
smaller, I believe,
cells are
but they have
small.
tons of cells.
Interesting.
And they have many other interesting things about them. But he debated between a few model organisms, and he chose C. elegans. I think it was a very wise choice. And he wrote that it's very easy to breed them. They're hermaphrodites, they're see-through, and all of that.
So I have another tweet here. It took more than a decade, but it's finally published. It started as a crazy idea, but after tons of hard work, we built it. We hijacked the endogenous secretion systems of Toxoplasma gondii, a brain parasite, to deliver active proteins to neurons.
Yeah, so this is
can
not.
you talk about this?
When you started reading it, I thought it was a funny tweet, but that's an actual tweet about the work that we published. So we published less than a year ago work on, which also took us forever to do and to publish, about engineering brain parasites to deliver proteins to the brain.
But Oded, what's the physiological relevance?
So the physiological relevance, maybe it can be a tool to deliver. So there's a big problem delivering drugs to the brain.
Mm-hmm.
There's the blood-brain barrier, which separates the brain cells from the rest of the body. And there are organisms that evolved to pass the blood-brain barrier and secrete proteins into neurons. One of these organisms is a single-cell parasite, which is called Toxoplasma gondii, which is famous for being a cat parasite, can reproduce sexually only in the gut of felines, and when it doesn't do anything bad to the cat, but if it infects a mouse, it gets the mouse. It changes the mouse's brain somehow, so it loses its fear of cats, and then the mouse runs towards the cat, and the cat eats it, and then the parasite can reproduce. It's good for everyone but the mouse. But it also infects every organism pretty much with hot blood, and including humans, and it gets to our brains as well. And when it gets to the brain, it has a few different secretion systems that it uses to secrete proteins into neurons. So it has one system.
It's able to deliver proteins into the neuron cells.
Yes.
Okay, wow.
So it has three different secretion systems. One of them is called micronemes, which is used to secrete proteins outside of the cells.
Mm-hmm.
Another one which is called rhoptery, which is like a harpoon that it shoots into the cell. And when it gets into the cell, it forms this vacuole. And out of this protective vacuole, it secretes dense granules, which are these vesicles that contain proteins that are secreted into the cytoplasm of the neuron. It gets cleaned from the rest of the body by the immune system, and it stays in neurons for the rest of your life. So what we did is everyone is trying to get rid of Toxoplasma because it can also lead to disease. But we engineered it as a sort of a way to deliver proteins to the brain. And the reason I thought this could be good is that, yes, it can lead to disease for sure. But normally, if you're immunocompetent, then you're fine. Then it's sort of mild flu, and then you're okay. Even if you're immunocompromised, there are drugs to treat it. And the estimates are that about a third of the world population is infected with Toxoplasma.
Wow.
So and most
we
of
know
us are
which
okay.
third?
We can check. I mean, in Israel, every woman that goes to have a baby gets checked for antibodies to Toxoplasma because it is dangerous to the fetus before it has a functioning immune system. But there are drugs that can be given. So it's a way to bypass the blood-brain barrier and secrete proteins to the brain, and it works. It works in cells. It works in mice. Whether it can be translated into a therapeutic, that's of course a
Is
million
it a way
times more
to develop
complicated.
to deliver an antibody drug, basically, for example?
So what you might be able to do, and we didn't do it, is to deliver nanobodies, which are these single chain antibodies.
Why not just because it can't produce antibodies, or?
Right. So this is very complicated. The process that leads to the generation of antibodies is complicated, but nanobodies can be genetically engineered and produced off plasmids even in cells. So you can, in theory, have the Toxoplasma produce nanobodies. We didn't do this, but it's in theory possible.
What did your work, what was included in your work?
So we showed it, we characterized which proteins can be delivered, what characteristics they have, and we also demonstrated it with a few different proteins, including proteins that lead to genetic changes in the recipient cells. So the thing that works very well is delivery of Cre, which is a recombinase that chops out or leads to inversions in the DNA. And we show that the Toxoplasma can deliver also in mice brains active Cre that not only is secreted to the cytoplasm, but goes to the nucleus and leads to a genetic change. It removed the Toxoplasma that we used, removed a stop cassette that prevents a gene from expressing. And once the stop cassette is removed, now GFP is expressed. GFP is a green fluorescent protein, and the brains shine in green. So this is something we showed. We also showed that the Toxoplasma can deliver CRISPR and TALEN, which are genome editing tools. But what we didn't show is that these remain functional in the cell. This could be an efficiency thing, perhaps, and something to improve in the future. But it was still, I think, a big thing to show that they can be delivered because these are very big proteins.
Can you, what percent of the neurons get affected? So is it like, 1%, 90%?
So it's a small percent of the worm, of the brain that gets infected, and this is something also to improve. But so also gene therapy when it started was tiny amounts, and it took 50 years to improve that. And I think that it can also definitely be improved with Toxoplasma. The trade-off is that you can increase the titer, for example, but you don't want to cause damage. Cells that get infected with Toxoplasma are also damaged. There are ways to go about this. For example, you can have Toxoplasma that are tamed and less harmful. This will enable you to...
You have to re-engineer them, basically.
You have to re-engineer them, but they are very easy to engineer. That's one... So that's something you can do. And another thing you can do is you can have proteins secreted from the parasites without the parasites infecting the cell, just outside, which is also something that can be done in theory. But I think taming the parasite and making it safe is the big challenge, and hopefully, like when we talked about the model organisms, this is a community challenge. So it's people we need to do it.
Is it easy? You were saying they're easy to grow, so you can grow them in vitro, or
you can grow them in vitro in cells. They're even studied. They resemble on very different organisms. They resemble Plasmodium, which is the parasite that leads to malaria. So many people study Toxoplasma to study Plasmodium just because they're much easier to genetically engineer.
Awesome. So couple more questions. One question is, you make jokes occasionally about like how long it takes to, get to any point of freedom and kind of like autonomy in academia. There's like one of them where you have I don't know this guy's name, but he's like a British royalty. He's maybe 70, and you have him labeled as like early career researcher. What's your take on the overall? I mean, science seems to have gotten to a point where it's very biased towards people over the age of 60 getting the grants. Especially, I mean, I think there's this crazy graph of like NIH funding, and it used to be like 5% of it would go to people over 66. It's like 30% now, and 30% used to go to people over under 35, and now it's like 2%.
Wow.
So it's a complete inversion.
Yeah.
Is this a good thing? Is it just because science has become very complicated, and so you need an extra 20 years to get to the point of like making good judgments?
It's very bad. I mean, it should be different. And I think part of the problem is it takes so long to publish. And also we're so...
So you actually bring this back, you actually correlate this back to the publication issue.
I think it's a publication. So science is not efficient enough because we're wandering in the dark, so it's difficult. And it takes us forever to publish. And if we don't publish in these high impact journals,
doesn't count.
It's not enough to get you a position.
Right.
So this is why I think we need unbiased, fast alternatives. The AI can review your paper in a second, and then perhaps it can accelerate your career. I think that's the problem. People are young, are early career researchers when they are 40 something, and at that point, some of them are frustrated. They go away from science. That's a big problem. In Israel, it's a huge problem. I think probably everywhere, but pretty much everyone in the lab has the alternative of going and working for tech companies, and they are more and more, tempted to do it. I see over the years. I have the track record. Track record is PI only 30 years. That's when I started my lab. I see more of my students now go to tech. Mm-hmm. The temptation is there. They can make 5 times as much money, and we are frustrating them with the process of promoting them and giving them independence.
Yeah, I think it's an interesting question how to retain the best people. I mean, I think a lot of people are not money motivated, but if you combine it with, it's like, okay, you're going to be in monastic poverty, and also, you don't really get to do the science, maybe later, like this kind of...
I mean, there are many advantages to working in science and to be in academia. We have freedom that people in tech don't necessarily have. But as you said, if it takes forever, and we also, don't let you study what you want and don't pay you well enough, and everything takes forever, then they will leave.
But how does that? Okay, but then you also complain that there's so much new literature, right? So how do these things? This seem like opposite problems. Like there's too many scientists, too many papers. Like it's happening too quickly, and no one even can read it all.
Because
so how do you?
I think we need to make better papers.
Okay.
I think we just and I think we need
fewer and better.
Fewer and better.
And what do you?
Sidney Brenner, the guy who started working with Elegans, he had the idea. He published this column in Current Biology for many years, where he gave advice. Some of it was humoristic and for scientists. So he said that he thinks that I don't remember the number. I think he said every scientist should be allowed to publish only 10 papers during their life. Get funding to publish 10 papers and that's it. But they should be good. Don't waste them. It's like Tarantino movies, How many did he say he'll do? 7 or 10? I don't remember. So it's like that. Maybe, I don't know. Probably that's not the best idea, but I think that we should make less paper, better paper, and publish them when they are relevant fast.
Yeah. That's a very, very interesting point. So the frustration with the deluge of literature is you're basically saying because this is the way that career advancement works, there's also just an overproduction of what might be. But I think, in the age of AI, brevity is becoming more valuable. Like it used to be valuable if you could generate blocks of text, like massive blocks of text. That's impressive. Like you have a high IQ, but now it's kind of coming out of a faucet.
But academia need to adapt for that because in academia it still counts. And but I totally agree. I agree.
Yeah, that's interesting.
But hiring committees or promotion committees, what do they do? How do they can't read the papers, don't have the time, and they don't understand it. So what do? They look at the heuristic of where it was published and the volume. So they tell you have to publish X amount of papers in your PhD to get tenure. And this creates this paper mill industry.
If you were a young person today, right? Like you're still young, but if you were very young, 20s, how would you approach your science career? Or even just life. Like what's your recommendation to kind of people similar psychological build to yourself? Like what would you do now if you were just getting into it?
You have to somehow not become addicted or to this publication of high impact papers because it ruins your life. You have to do just good science that interests you. Try to create a setup to do it. And I think that is the best advice. So don't be a slave for that. You can always do something else. You can always do something else. And I think that the biggest risk, so people trying to plan their careers, where will they do their postdocs? Where will they go? What will they study? And they try to make these careeristics, strategic planning. And it's a very bad idea. The number one reason that will get you out of science and leave the profession is you're just not interested anymore or you're frustrated. So if you stay happy and you study what you like and just focus on enjoying it, and it's very hard because there are many forces working against it, and perhaps it's okay because as I said, it is a loophole in the world that people just tell you, study what you want and have fun. So I can understand why the universe is resisting this, but I think that should be definitely the goal, and it's the number one recipe for success.
This is maybe a sensitive question, so you don't have to answer if you don't want. I can just cut it out. But like doing science and this kind of, just like free exploration and things that really don't have, it might have some practical significance, but not yet, right? And it's certainly not the main reason probably that you do it. It's what's it like doing it in Israel, which is basically, I mean, on and off a war zone, right? And sometimes with like absolutely distracting, insane things happening, right? Like how does that impact your, mindset about it?
It's a very good question. So yes, it's a crazy place. It's a difficult place, but this is, where my family is, my friends are, and I love it, and I want to stay here with all the problems that it has. It's very disruptive for science in recent years. I have students recruited to the army. I have all kinds of disasters occurring around me. It's hard. There are also difficulties elsewhere.
I mean, where's in this building? Like when there's a siren, where do you go? Where's the bomb shelter?
To the stairs, it's not really protected. Yeah, it's insane. On the other hand, the last couple of years, and we've been like that forever, but the last couple of years are special. They're especially hard.
Mm-hmm.
But I think that it gives you proportion a little bit. So, during the worst times, I couldn't care less if my paper is accepted or not.
So
and I think so. So that is good. And I think the perspective is important.
Mm-hmm.
But it's not easy. But now there are trouble everywhere. Also in the U.S., it's a mess. And
in the U.S., the biggest problem people experience in their life, maybe their whole life, is like a food delivery being late or something.
No, I'm talking with the scientists there now. And I think in Israel, the government didn't reach the academia yet. I mean, they tried a little bit, but it didn't work. But it can definitely happen. It can definitely happen.
Well, but I mean, look, so I think I have a pretty unbiased perspective because I don't work in academia, but I've spent some time in it. And, I really enjoyed my time. Like every moment I spent in universities, I had nothing but fun because I never expected a career in academia and I never had like a lot of the issues. Okay, a lot of my work didn't get published, whatever, like some of it is only on archive. All right, whatever. Like it's not. And so I had only had fun. And, when the funding cuts and all these things, I mean, people seem to have this like duality where on one hand, they're super frustrated. Like this system is just terrible, right? Like there's so many problems with this system. It's wasting everyone's time. There's all this negative, honestly, right? Like people are not, they don't like it. And then there's like a kind of like a people from the outside saying like, well, we don't even want to pay for it anymore, And there's this like, kind of like a duality where they're like, wait a minute, we should still pay for this even though it's terrible.
No,
so there's kind of like
But I think the question is what is terrible? Yes, of course, there's room for improvement and for more efficiency, obviously. And I'm talking about it. But the question is how, the how.
Yeah.
And at the moment, the how doesn't seem good at all.
But I think, doesn't it give you hope at least that I totally agree that just randomly, it's like if you have a broken car and you just start hitting it randomly with a hammer, it's not going to start running,
and
right?
you will just remove two wheels.
Yeah, exactly. Like it's not useful to do like just increase entropy is not helpful, right? But I do think that there's a certain kind of like, when the computer is not working, sometimes it's so complicated that turning it on and off is like the only option. So I do think that there's kind of a thing where it's like if you admit that it's not the most functional system, there is like maybe a benefit not to just smashing with a hammer, but like in some way having some type of reset mechanism where it's not just we're just doing the same thing we did, but taking like very incremental steps, small changes.
I mean, I think we need to think how to improve it. I'm thinking, as you heard, I'm really thinking about it. I think it's one of the things that interests me the most. But I think it should be done in a smart way. There's no shortcut.
Oded, thank you very much. This was awesome.
My pleasure.
Thank you
totally
for taking
my pleasure.
the time.
Thank you.